Thank you. Well, good. Yeah. Thanks, everybody, for your attendance. And it's a pleasure to speak to you all today. I think these seminars where we've heard from our own faculty have been really useful for me. I often think that I know vaguely what people do, but never know their details. And so maybe some of this can be continued after the post-COVID period, whenever that is, and we can hear from a few of us through the year, as well as obviously getting outside speakers. So what am I going to do today? I'm going to discuss the rationale for looking at sex differences in medical outcomes, give you a little bit of background information. So even if you're not interested that much in stroke, you can come away from this talk with a little bit more understanding about why we care about sex differences. I'm going to give you a brief review of the historical approach to dealing with sex in biomedical research, including previous data on sex differences in clinical trials participation. And then I'm going to get into the specifics of a meta-analysis for looking at sex participation, sex differences in participation in acute stroke trials that we recently published on early this year, and wrap up with some discussion of what causes these differences and what can we do about them. Okay, so no conflicts of interest. The disclosure I want to tell you, perhaps with some warning, is that none of my work related to sex differences in stroke has been grant funded, despite the fact that I've submitted several grants over the years to try and get funded in this line of work. So I'm not presenting this work as a doorway to a fruitful, highly enriching research environment. It's tough to get research on this stuff. So a good question to start with is why study sex differences as an academic pursuit in the first place? There's plenty of good and entertaining books about how men and women differ. These have been number one bestsellers on the New York Times list for decades now. And, we know men and women are different. We understood that ever since we were boys and girls. So what's the point of studying this? So I hope to be able to answer that question today. I really, didn't start off thinking I would, start studying sex differences in stroke outcomes. It was really a kind of a fortuitous thing. I was sitting on a large amount of registry data that I'd collected. This is about 18 years ago, and a student who was here, Julie Gargano, was working with me at the time, and I said, well, what do you want to look at? She said, well, let's look at women. And I was like, sure, I like women, let's do that. So we kind of started analyzing the data according to looking at difference between women and men, and published a couple of papers on quality of life and differences in outcomes, particularly functional recovery post-stroke. And we thought they were pretty interesting. And we looked in the literature and we didn't find a lot of other studies on them. And it was from that I tried to submit several grants to study sex differences in stroke outcomes specifically, with not much luck. And during that process, I decided to write a review article about sex differences in stroke. And because I was getting challenges from reviewers saying, well, this is really not that important, and it's all due to confounding, and why bother? So this is a paper I finally published in 2008 in Lancet Neurology. And, most papers I publish just sort of disappear or percolate in the background. But this particular paper has taken a life of its own. It's now got anywhere between 650 and over 1,000 citations. It's probably the most widely cited paper about sex differences in stroke. And it's a narrative review, the very lowest level of evidence. But certainly the timing was right, and it caught a real upsurge in the interest in looking at sex differences in stroke. So since this time, people have decided that I must know a lot about sex differences in stroke, which has forced me to continue to keep up in this area since, I get once in a while invited to talk about this topic. So let's give a little bit of background on sex differences. Let's have the conversation about sex, and particularly the distinction between sex and gender. I want to talk about framing, which side of the coin to look at, and then a quick review of biological versus genetic versus the cultural origins of these sex differences. So let's talk about the difference between sex and gender. So sex refers to a set of biological attributes that are associated with physical or physiological features, including chromosomes, gene expression, hormone function, or reproductive and sexual anatomy. And it applies equally to humans, animals, and cells. It's a classification variable, traditionally specified as male or female. It's determined by innate biology, and it tends to be the primary focus of biomedical literature. You'll see more studies referring to sex differences than you will gender differences. So what do we mean by gender? Well, gender refers to the social construct of the roles, behaviors, and identities that females, males, and gender diverse people take on. And by people, I mean humans. So gender is specific to humans, whereas sex can refer to animals or cells or whatever. And gender is often incorrectly specified as dichotomous, male or female, but as probably we all appreciate, it exists on a continuum or a spectrum of identities and expressions. And it's important because it affects how people identify and express themselves and how they interact or perceived by others. It's, of course, influenced by an environment and environmental experiences. And this seems to be the primary focus of social science literature. But the bottom line is everybody's very confused about sex and gender and the distinctions. It's not that difficult, but in the medical literature, you'll see plenty of papers that in the title refer to gender differences that could easily have said sex differences. And in none of the papers, including the papers I write, do I ever sit down and rationalize why I'm referring to sex differences rather than gender differences. But I think we should, and whatever term you use, it would be good to understand your rationale for that. Do you think that what you're looking at is more culturally and socially determined, or do you think you're looking at a situation that's more biologically determined, or are you looking at the intersection or the interaction of these two features, which could be done, although it's a lot harder to do? Things, back in the day, everything was black and white, and we talked about men and we talked about women, and that was it. That was all as complicated as it got. And then, but things, as you realize, have gotten a lot more complicated. This came out in March of this year, and it's the change in the self-classification of gender that MSU will be using to classify people or have them self-classify. And this was the minimum list here, which prompted me to send an email to the people who sent the email out saying, can you give me some definitions of this? And I got a somewhat terse response saying, well, you should look at this glossary of terminology. They gave me this link here at the bottom. So, and if you go on that, it's pretty fascinating reading. There's at least 60 terms that have something to do with gender identity, sex, sexual preferences, sexual identity, et cetera, et cetera. So, the medical literature is probably never going to catch up to this new way of looking at things. And just to make the point that we are still working in the binary world of you're either male or female in terms of the sex difference. The other thing I want to say about sex difference work is it's also about framing. And the point is, which side of the coin do you want to look at? Because a sex difference is a contrast between one sex and the other. And you have a choice to compare women to men, with men as the denominator, or you could compare men to women with women as the denominator. And so really, sex differences is studying the two sides of the same coin. And which contrast, which coin, face of the coin you want to work with depends on what message you want to send and what your context is. And in medicine, I think it's generally true that most sex difference studies are framed in terms of studying women relative to men. But of course, you could easily turn that around, studying men compared to women. I can just tell you right now, you'd have a lot harder time getting the same study published, framing it as men compared to women as women compared to men. So bear that in mind if you start doing sex differences work. And similar to that last point I made is, I call this, it takes two to tango. And that means, what I'm trying to get at this is a sex difference is an explicit relative comparison. And so any observable sex difference, whether you want to look at a ratio measure, as I'm showing here, or a difference measure, as I'm showing here, it says as much about the denominator as the group as it does about the numerator, for a ratio at least. So, sex difference could be what's more reflects of what's going on in the bottom group as opposed to the top group. And as an example, you think about trying to quantify how much do people drink and smoke. And so when you look at the data, you could make the argument, well, it's women who drink and smoke too little, or is it that men who drink and smoke too much? And of course, the answer to that depends, because everything is relative. So just remember that any sex difference is sort of an arbitrary way of deciding which is compared to which. So what are the origins of these sex differences? Are they biological, genetic, cultural? Sort of a seminal publication on this was this Institute of Medicine report back in 2001, exploring the biological contributions to human health. Does sex matter? This is where it really laid out on behalf of really the NIH why we think there should be a renewed focus, or we should begin to focus on looking at sex differences rather than ignoring them, which has been sort of going on to date. And there's been, several good articles and journal publications about this. Whatever area you're working in, you can be assured that someone somewhere has talked about sex differences in whatever outcome disease you're working with. And a paper I've been using quite a bit lately is this one review that came out in Lancet last year, Sex and Gender Modifiers of Health, Disease and Medicine, which is also a good place to start if you want an introduction to the topic. So origins of sex differences. Just make the point that sex differences can be studied at all levels. You can study this at the individual, the whole organism level, whether it's animal or human. You can also study this at the cellular or even molecular level, looking at female cells versus male cells. And then you can step back from all of that and look at sex differences from a societal or true population level. And we think about biological differences for these sex differences can result from obviously the sex steroid hormones, estrogen versus testosterone, but it's more than that, obviously. It's also differences in lifestyle and health behaviors. And then specific to women, there's the role of reproductive health and pregnancy that carries through lifelong benefits and potential harms through the life course. And then in terms of genetic differences, we have these fundamental genetic differences of XY. On the sex chromosomes, you either get XY or XX. And what these result in sex-specific gene expressions related to whether you have 2 copies of X or 1 copy of X and a copy of Y. And then there are epigenetic effects, for example, the interaction of genomes with this testosterone surge in males, or actually in females. An interesting issue comes where there's a random inactivation of one of those 2 X chromosomes in females, or there normally is. And which one gets the lead, it can have an implication for epigenetic effects. So those are genetic differences, biological differences, and finally the sort of cultural or gender-based differences. And these are affected by country, region, age, race, ethnicity, education, socioeconomic status, religious status, and the time period you're looking at. And so you have all of these 3 things working together. And they work together or they interact to influence a whole range of health statuses and outcomes. So this whole list, you can all appreciate that, yes, sex or gender impacts behavioral norms, the degree to which you follow healthy lifestyles, preventative behaviors, your risk exposures, whether they're environmental or occupational. The propensity to take risk-taking behaviors is usually much greater in men than it is in women. Health-seeking behaviors are different between men and women, and the utilization of healthcare services is different. Then healthcare interactions differ by sex and gender. The perceived experiences differ by sex and gender, and shared decision-making differs also. And then we haven't even gone into the sort of routine epidemiology of differences in disease prevalence, incidence, and mortality. there are differences in response to treatment and prognosis. And then if you get into patient-reported outcomes, there are quite big differences in sex differences in quality of life, well-being, satisfaction, and things like that. So I think we all have an appreciation for, yes, sex, gender matters across healthcare. It's pretty easy in whatever area you're working in to find pretty obvious differences. They're probably so obvious that you haven't really thought about them, but they're there, and they may be of relevance. And then this idea that sex and gender intersect to impact on health is shown in this diagram, where we have the biological basis of sex with sex chromosomes leading on to difference in hormones, combined with the gender constructs, which are sort of driven by society, and the interaction of those epigenetic interactions leading on to changes in behavior and interactions with health-seeking behavior, as well as disease manifestations in response to treatment. So these two things intersect, interact, however you want to describe how these two things work together. However, it's worth taking a step back and say, well, despite all these big differences between men and women, the big picture in terms of disease burden is pretty similar between the sexes. This is a depiction of the top 10 causes of death in the US for the United States. And, 8 of the 10 causes are the same between males and females. Heart disease and cancer being the number 1, number 2. And then, there's a little bit of shuffling between stroke, injuries, Alzheimer's disease, type 2 diabetes. But, there's a lot of similarity, which is often the case. We look at sex differences. There's more similarities between the sexes than there are differences, which is something we want to remember. Okay, let's talk a little bit about the historical approach to dealing with sex in biomedical research. And so looking at the basic sciences, I wasn't really aware of this until I started reading up on this. And some of these differences, historical approaches are pretty egregious, for sure. So there was an assumption made early on in a lot of basic science that, male and female cells were essentially the same. Their response to stimuli, to manipulations are going to be the same. And it was also assumed that, females could produce an unnecessary amount of variation in these basic science studies. And so most animal research was done on male animals only. A couple of studies have shown that, 80% of the research in given basic science areas was done on male mice, male rats exclusively. So, many a young lab technician started off in life getting rid of the female mice because they weren't needed. Right, we're just going to do this experiment on males, and so there was a lot of work done to get rid of the female mice or the female animals. And then in clinical research, women of reproductive age were often excluded from participating in clinical trials, which is the topic I'll be talking about today. Because of vague, undefined concerns, but if push came to shove, there would be concerns listed about fetal risks and also the effect of hormonal variation impacting whatever outcomes they were looking at. Again, unjustified, but used at the time. And also, I think it's fair to point out that a lot of the early clinical and epidemiological research tended to focus on diseases that impacted men more than women. An example would be, early studies of lung cancer, cardiovascular disease in particular, focus on framing and, for example, occupational diseases. And this was widely interpreted as a form of sexism. And to a degree, it probably was, but maybe also it was just the focus of looking at the big disease burdens at the time in the 50s and 60s. And it wasn't until then, 1993, that NIH first introduced policies to ensure the inclusion of women in clinical research and of female animals and female cells in basic science research. And this gets to this policy of sex as a biological variable, SABV, first introduced in 1993, updated a couple of times, and most recently in this article by Janine Clayton in 2018. And the sex as a biological variable statement from NIH basically said NIH-funded research should consider the effects of sex as a biological variable in all of its stages, including study design, analysis, and reporting. And if you don't consider sex in your analyses, in your study, you've got to say why. You've got to say why, or you should say why sex is an irrelevant issue here, and why it shouldn't have an influence on particularly the design and analysis of the study. The goal of this SABV statement was to increase, obviously, the representativeness of research, so it's generalizable to both male and females and the population as a whole, and also to improve the reproducibility of a lot of research. As you know, that's been a big problem, particularly for basic science research. And eventually this led to the development of the SAGE guidelines, the Sex and Gender Equity Research Guidelines, which is really a reporting guideline telling you how to write up studies about sex or gender difference. And so that's used by technical editors when we're looking at studies that are sex or gender based. So you should know about those. Okay, so a brief review of data on sex differences in trial participation. So this is well, let's stop right here for a second. So is there any? Let me stop for a second and just see if there's any questions or thoughts on anything about this background information that I gave on sex differences and gender differences research. Fair enough. That gives me a time to slug a bit of tea down, so that's fine. All right, well, maybe you can come up with some questions for me at the end, but let's move on to the specific topic of today, which is to talk about trial participation of men and women in randomized clinical trials. And the fact is that the underrepresentation of women in trials has been a commonly recognized problem, reported in oncology trials, in dementia trials, in cardiovascular disease trials in particular. Most of the work has been done on cardiovascular disease, where the estimates of the under-enrollment of women on an absolute scale vary anywhere between 9 and 18%. So between 9 and 18% deficit of women in cardiovascular disease trials. And this has been shown sort of repeatedly over the last 15 to 20 years since this has been looked at in earnest. And for acute stroke trials, we also know that women on the surface seem to be underrepresented in trials. So 40% of National Institute of Neurological Diseases funded stroke trials, so the stroke NIH Stroke Center, 40% of participants in those trials were women, and only 40% of trials registered on clinicaltrials. Only 40% of participants were women in trials registered on clinicaltrials.org over a 15 to 20 year period, which points to a problem, although these studies didn't account for the underlying prevalence of stroke in the population from which the trials were done. And we needed a more detailed analysis of the reasons behind these differences so we can kind of figure out what to do about them. So this led to the study I'm going to talk to you about today, which is a meta-analysis. This was published in JAMA Neurology earlier this year. So basically, I want to describe the evidence for sex difference in the enrollment of these trials, these acute stroke trials, after accounting for the sex ratio in the underlying population. We want to assess associations between key trial eligibility criteria and whatever disparity estimates we find. And then we're going to wrap up and discuss potential causes and solutions. So, this is the work we did published earlier this year. We're all riding the coattails of the eponymously named Mr. Brent Strong, who was my undergraduate student at the time, who worked on this. Also, Dr. Lacampos helped us, and a couple of medical students, Julie Pudar and Mutaza Hussain, are medical students who helped us over the couple of years that it took us to do this, and then also some collaborators in Alabama and in Australia. So this is a straightforward meta-analysis. We kept things really pretty simple. We only looked at Medline for acute stroke randomized trials published over a 10-year period. And we limited it to 9 major clinical journals because we were wanting to focus on clinical trials that would have a meaningful impact on clinical practice. And if you have an important trial that you think is going to impact clinical practice and it's in the English language, you're going to publish it in one of these 9 trials. Very few, 9 journals. Very few other journals publish major clinical trials in the area of stroke. So that made our job easy. We just had to look at these 9 journals. We did a standard search for stroke randomized trials. Things are never quite as simple as you think, because then we discovered, even though we looked at every permutation of acute stroke trials, we'd actually missed quite a few hemorrhagic stroke trials. So we had to go back and redo the search, adding in terms for cerebral hemorrhage and subarachnoid hemorrhage, which are a different subtype of stroke, apparently not captured in the mesh term stroke, which is something we learned. Anyway, that's how we found the corpus of studies. We assessed eligibility eligibility for manuscripts to be eligible. They had to meet the following criteria. So the studies had to present the final primary results of either a phase 2 or phase 3 randomized trial with a planned sample size greater than 100. So if it was a secondary analysis, if it was analysis that uncoupled the randomization, we weren't interested in that. Small phase 2 exploratory studies we didn't include. The trial populations could include any type of stroke patients. You should know there's ischemic stroke, which is the major component of stroke, but there's also hemorrhagic strokes, which can be intracerebral hemorrhage or subarachnoid hemorrhage. And there's a thing called TIA or mini strokes. We included all of those. We included any acute treatment, whether it's pharmacological or non-pharmacological, initiated within 1 month of onset of the stroke, with a comparison to a control group. It could have had an active treatment. It could be placebo-controlled, sham-controlled, or just receive usual care. We excluded diagnostic, education-based, and quality improvement-based interventions. And we followed the PRISMA guidelines to report in systematic reviews and meta-analysis. In terms of screening, 2 authors, Brent and Julie, independently screened these titles and abstracts for potential hits. They then, the potentially relevant citations then underwent independent full text review to determine final eligibility. Then we dual abstracted data out of these trials, out of the eligible papers, and then all discrepancies were resolved by myself, either in terms of eligibility or data abstraction. And then we abstracted data on the major trial characteristics and the 3 key eligibility criteria. And again, that was done in duplicate. So here's the data that we abstracted from each study, the ratio of the number of enrolled women to the number of enrolled men, where these clinical sites were, what was the period of enrollment, what was the geographical location, how many people were enrolled at each of the sites in each country, in each region, details of the particular intervention, the experimental intervention, the type of intervention. I'm going to be talking about IVT and EVT. IVT is intravenous thrombolysis. It's a drug that's injected into the vein to remove the clot, which is likely to be causing the stroke. And EVT is endovascular therapy, which is the use of a device that's threaded up into the brain, into the vessels to remove a clot. So those are 2 major types of interventions for stroke trials that are very important to stroke patients. We also abstracted what the primary endpoint was, what the pre-planned and final sample size, was there any involvement of industry, what was the age of the participants, what was the distribution of stroke types, were any women involved in trial leadership? So was a woman the first author of the paper or listed as the senior author? Then we abstracted patient-level eligibility criteria, since we're going to use these criteria to look at what could have influenced the proportion of women, enrollment of women in these trials. So we looked at age, eligibility criteria related to age, stroke severity, pre-stroke disability, time from onset, eligibility for specific medical treatments and qualifying comorbidities such as hypertension, diabetes, and AFib. Now, the first outcome measure we took from these abstracted data was the most simplest, and that was the PPW, or the proportion of trial participants that were women. This is simply the fraction of study subjects that were women amongst all the study subjects in the trial. So PPW is simply the proportion of women in that particular trial. As an example, Mr. Clean, very important trial, stroke trial, conducted in the last 10 years, enrolled 208 women out of a total of 500 men and women for a proportion of 41.6%. Okay, so that's their PPW. Okay, and we're going to abstract that and calculate that for every trial we find. But I also mentioned earlier that previous studies hadn't really accounted for the underlying expected ratio of men to women in strokes that are occurring in a given population. And so that's what we wanted to do. And so what we wanted to do, we next matched each individual trial to the Global Burden of Disease database for 2017. And we did that according to where each trial was conducted, either a specific individual country, a specific geographical region, or potentially a region within a single country, if the country was large enough, such as China or the US. So we went into the Global Burden of Disease and abstracted the stroke incidence data from whatever available population there was. And specifically, we abstracted the number of incident strokes in men and women. So the numerator of the stroke incidence rate, the global burden of disease reported for any given population, okay? And we also abstracted the 95% uncertainty interval. They phrase their variance in terms of an uncertainty interval rather than a confidence interval. And we use the middle of each trial, the middle year of each trial's enrollment period to go find the global burden of disease data, okay? And this data was then used to generate the PSW, which is the proportion of strokes occurring in women in that underlying population, okay? So how do we calculate that? It's simply the number of women we stroke in the population divided by the total number of strokes in the population. Again, this is coming from an incidence study or an estimate of the incidence study, but it's abstracting the numerators and not necessarily the incidence rates, okay? So as an example, Mr. Clean, the same study from Holland, enrolled acute stroke patients over this 4-year period. And so in 2012, we used the global burden of disease to estimate that the proportion of women amongst all stroke events was 50%. So there were 50% of stroke events in the population in Holland were women, whereas only 40% or 42% of strokes enrolled in this particular trial were women. Okay, so remember that, because we're going to put those two pieces of information together to look at, finally quantify the enrollment disparity difference, or the EDD. Okay, the EDD was first used in assessment of lung cancer trials, and so we made a modification to it. But essentially, for each individual trial, we calculate the difference between the proportion of women in the stroke trial itself minus the proportion of women in the underlying stroke population, the PSW. And we're going to do that, calculate this EDD for every individual trial. So a negative EDD indicates that women were underrepresented in the trial relative to the frequency of stroke events in women in the underlying population. So negative EDD, under-enrollment; positive EDD, over-enrollment. An EDD of 0 means the trial enrolled exactly the same proportion of women as was found in the underlying stroke population. So for Mr. Clean, I remember I gave you that 41% proportion of strokes enrolled in the trial compared to just shy of 51% in the underlying population, gets an EDD of negative 0.092 or negative 9.2%. Okay, so on an absolute basis, women were under-enrolled in the Mr. Clean trial by 9.2%. And what we want to find out is why is that and what contributes to that. Quantifying the imprecision of the EDD, we calculated standard errors. Won't go into the details, but we used standard methods for proportions, and then we had to do some bootstrapping and some fancy statistics that I appreciate Dr. Della Campos helping us with to estimate the standard error of the PSWs, the proportion of strokes in women in the underlying population. So we generated that data, we abstracted the data, generated the PPWs, the PSWs, calculated the EDDs, and now we want to do a meta-analysis on those EDDs. To do that, we're going to do a random effects meta-analysis on the EDD using Stata. We're going to conduct subgroups across different characteristics of these trials by geography, stroke type, sample size, industry involvement, intervention type, the type of treatment that was being tested, and whether women were in a leadership role. Then we're going to do some sensitivity analyses. We're going to explore whether the global burden of disease data was causing any bias. And we're also going to re-express the data in terms of a ratio measure rather than an absolute difference measure. We're also going to do something called meta-regression. We're going to use a random effects multivariable meta-regression to investigate associations between trial eligibility criteria and study-specific EDD estimates. So the meta-regression is done on trial-level characteristics. So each trial is its own observation in this multivariable analysis. And we're going to study specific eligibility criteria, including what was the highest age that they would allow participants to have. Was there a limit on the amount of pre-stroke disability a patient could have to get into the trial? What was the longest permitted time period from stroke onset to clinical arrival and enrollment to treatment? What is the range of stroke severities they included? Is there a requirement for the patients to be eligible for this IVT or EVT treatment? And is there a requirement for the presentation of hypertension, which was the most common comorbidity? So we're looking at these various eligibility criteria to see which ones seem to impact the variation or captures the most, explains the most variation in the EDD across these trials. So getting on to the results then, we found 1,529 hits, eventually finding 155 eligible trials, as shown here. These 115 trials randomized 121,000 participants, so a lot of subjects, 43% of which were women. 23 trials were from Americas, 22 from Asia, 48 from Europe, so nicely spread across the globe. 80 trials enrolled acute ischemic strokes or TIA patients. 19 trials enrolled hemorrhagic stroke patients, and 16 trials enrolled both. And 19 trials tested EVT, 19 IVT, secondary prevention, a few surgery. And then the biggest category was 61 trials, which were in the other category. Anything else, most common being some sort of neuroprotective treatment was being tested. These data are all what's shown there. So what do we find? We found that the median proportion of women stroke cases in the trials was 45%, with a range of 22% to 79%. In terms of the underlying population, we found that the average PSW, or the median PSW, was 50%, but the range was quite wide, anywhere from 41% to as high as 67%. So that's the proportion of women in the underlying stroke populations. And our random effects pooled average EDD was negative 0.053 with a confidence interval of negative 0.065 to 0.04. What does that mean? It means the proportion of women enrolled in acute stroke trials was 5% lower relative to their frequency in the underlying stroke populations. This estimate of the absolute difference was precise, as indicated by the confidence interval, and also is significantly different from the null value. We did subgroup analysis because we found substantial between study variability. In other words, this enrollment disparity is now consistent across all trials. There's a lot of variability, The biggest source of variability was for subarachnoid hemorrhage, type of hemorrhagic stroke, where the EDD was positive 0.12, so women were over-enrolled in subarachnoid hemorrhage trials. And the biggest under-enrollment was seen in the secondary prevention trials. So this is shown here in this forest plot. Here you can see to the left of the horizontal line, whether you can see my mouse, but everything is shifted to the left, which favors men, because you've got a negative EDD, so more men are enrolled in by most of these subgroups. There in red at the top is the 5% disparity favoring men. Here's the positive over-enrollment of women in subarachnoid hemorrhage, and here's the largest discrepancy, a 12% discrepancy, absolute difference amongst the secondary prevention trials, again, favoring men. Okay, so that's the subgroup analysis, generally finding very consistent results apart from those two highlighted areas. In sensitivity analysis, what we did here is we had 49 trials where it was possible to swap out our global burden of disease estimates of the underlying proportion of strokes in women with a specific study, population-based incidence study conducted in that particular country. So we got rid of the global burden of disease since, there are some questions about its reliability. There's some extreme skeptics out there. And we replaced that global burden of disease with a country-specific estimate. And when we did that, we found that the EDD, the overall ED, actually increased. It went to 0.07, so 7%, as opposed to 5.6% when it was just based on global burden of disease data. And then when we just converted this data from a difference measure to a prevalence measure, a ratio measure called the participation to prevalence ratio, the ratio is 0.89, meaning another way to frame the same data is to say participation of women in trials was 11% lower relative to the representation in the underlying stroke population. Okay, so our meta-regression basically found two things impacted enrollment of women. Trials that used an upper age limit of 80 years was associated with a 6% decrease in enrollment. So a trial that said we're not taking anybody else above 80 years of age into our trial resulted in a 6% fewer women being enrolled. And then encouragingly, we found that these IVT and EDT-based trials were associated with a 3% increase in enrollment of women, which is, again, was a surprise. So the biggest conclusion from a meta-regression was the fact that we've proven that an upper age limit was detrimental to the enrollment of women in trials, and this is these data in table format in the paper itself. So some limitations then, the over-enrollment of women for the subarachnoid hemorrhage trials may have actually been a reflection more of the bias in the global burden disease data than in the trial data, since subarachnoid hemorrhages are very rare and yearly global burden of disease estimates vary widely because it's just so rare. And the usual concerns about meta-regression are a form of ecological study and subject to residual confounding. And obviously, we only included trials that were published in major journals in English, which would limit generalizability of the stroke trials in countries and regions and other languages. So again, the main findings is that 5 percentage points, women were underrepresented by 5 percentage points in these trials. These differences were robust in subgroup analyses into sensitivity analysis. Just to make the point that this 5 to 6% absolute decrease may not represent an overt disparity due to sexism, ageism, or other biases. It needs to be explained. It's small enough that it could easily be due to some hidden or confounding factor. But regardless, any discrepancy adds to the concern that trial results are not representative, may have poor generalizability. More broadly, discrepancies also add to the concern about the fairness and equity of research, of the general research enterprise. So it's not something we should favor seeing even a small difference. Just to make a point, the our difference in stroke trials is actually less than observed in the other cardiovascular disease studies, which I've already said are often somewhere between 9 and 18 points % against women participation in trials. So this is a common problem, particularly in cardiovascular trials, so we're not quite as bad as the cardiologists. The other point was that the issue about the effect of excluding patients above 80 lowered enrollment of women. This is entirely predictable. We know that women with stroke are about 4 years older than men at the time of the stroke. But, look at this age distribution in this figure here on the right-hand side. There's dramatically more women in those older age groups, 80, 85, such that if you limit enrollment at 80, you would exclude 19% of men, but 44% of women. This is data from a German stroke registry, but it's widely replicated. So there's just this huge number of stroke events in women 80 and older. So any restrictions around these older age groups has a big impact on women. So again, this age limit is probably not ageism. It's more that age is probably being used as a proxy for some other thing, concern about frailty, fall risk, medication side effects. So what is the solution? Well, we think we should get rid of the age limit and replace it with whatever valid risk score. So if you're worried about frailty, there are risk scores to quantify frailty. If you're worried about cognitive decline, then screen for cognitive decline. It's not really fair or appropriate to just use a straight up age criteria anymore. The findings that there was no sex differences in IVT use was in contrary to several prior papers, including our own work, which has consistently found discrepancies in observational data about the use of TPA in women. So quickly, I want to just sort of talk about the full potential mechanisms for why women might have lower participation in trials. It could be that stroke patients did not receive medical attention. It could be that the stroke patient was not screened or approached by research staff. It could be that the stroke patient refused or declined participation. And it could be that the stroke patient was excluded because of trial eligibility, which is what we studied. We only studied number 4 out of these 4 possibilities. Okay, so it is important to know that 10 to 15% of stroke, it is estimated, events don't get hospitalized, and that this disproportionately affects the elderly and women, particularly those in nursing homes and institutionalized care. So that's recognized. So that's one possibility why you might see less women events than you would see in the stroke population as a whole. Also, it's the case that not every patient is screened for enrollment in trials, and there are ad hoc decisions often made about who to approach and assess for eligibility. This is probably the best example of where implicit bias could be active, where someone who's involved with trial enrollment looks at a patient from afar and goes, "Nah, they wouldn't be interested." That's a common problem. And especially a problem because trial pre-screening procedures can vary a lot between trials, and they're often poorly defined. And if you start looking at the top of the consort flow diagram, when you start looking at trials, notice the variability in how the top of the consort diagram is organized and what gets listed here. And here's one example where it says straight up, the total number of patients assessed for eligibility is not available because screening log data were not available. The point is, we don't really know. We don't really have processes in place to ensure that everybody is screened and given the opportunity to enter a trial. And certainly the consort trial criteria right now don't do a good job of forcing trialists to actually collect that data and present it. Now, once you actually do present a trial to a patient and tell them about it, then you have the problem of does the patient refuse or could decline? And it's certainly true across multiple studies that some are simulation-based, some are observational-based, but it certainly seems to show that women are more likely to refuse to participate in a trial. They tend to be more risk-averse, have less confidence or belief in the potential benefits of the trial. And it may just be that they're smarter than men and know that, hey, you're trying to experiment on me? Forget it. Go experiment on somebody else. So it may be just they realize that actually what a trial entails, which is we're going to experiment on you. So, and then, also what's been noticed is that women will identify that the burden of trial participant affects their willingness to participate in trials. So women are much more likely to list challenges associated with family caregiving, transportation, the costs. This is a particular case in older women, particularly the case in rural women. And also, this is backed up by the fact that there's been studies to show if you provide monetary incentives to participate in a trial, these are more effective in women than in men. And so, on the face of it, women are perhaps more bribable. So they don't want to participate, but if you give them enough money, maybe they will. So that is one solution, is to provide more monetary incentives. And then the issue about eligibility, once you do participate in a trial or willing to participate, then are you eligible to be in that trial? And as we found, particularly older age is a big factor. And other cardiovascular disease studies have shown that older age and limited life expectancy criteria will decrease the enrollment of women. And that basically the older you allow a trial population to be, the more, the higher the rate of women will be enrolled. So in other words, if you remove any age limitations, you will enroll more women into your trials. So again, making the point I made earlier that we should probably do away with those criteria. The other point made about trials, about sex-specific data, is we should report trial data by sex-specific, using sex-specific data. So we should always stratify results and provide sex-specific data for men and for women. We looked at that in the same body of trials and found that only 37% of these 115 trials provided sex-specific results. So we have a long way to go to meet those SAGA guidelines on reporting that all trials should provide sex-specific results. That was a little rapid run through towards the end. I apologize for that, but I've got a few minutes for questions and any thoughts. So opening up to the crowd.